lets people know, no, rules.
Okay. Welcome to our second, I was going to say annual, but it's actually the same calendar year, so that probably doesn't count. Our second Biotech panel. If you haven't signed in, definitely see me before we do the raffle, because I want to make sure we get you credit for being here today, and also to give you the raffle ticket. We have some great prizes tonight. We've had a biotech major and graduate program for 2 years now, and I think this room is a testament to how much it's grown and how much it's thriving. And just to talk a little bit more about it, I want to introduce Dr. Siddharth Saman.
Hi everyone. So I just wanted to kind of talk an overview on the biotechnology program at Elms College, and the information is really on the website. There's a good information that you can see on the courses and the degrees that we have. We have an undergrad in biotechnology, and there are a variety of courses that you can actually pick and choose to work on achieving a bachelor's degree. We also have a minor in biotechnology, and also the information in the courses are online. And we have a master's in biotechnology, and the master's program has actually picked up, and and we have now roughly about 8 students in the program. There's about 4 more that are basically applied for the spring. And in regards to the master's in biotechnology, we have actually 2 options that students could take. So the first option is a degree completion option that focuses on courses. So you only take courses with few capstone projects here or there. And that's 32 credits. And then the other option is actually research option. And that one you take 12 credits of courses and 20 credits of research. And just really quick is that the Masters of Biotechnology, one of the things that we stand out from other colleges is that we are small, and that there's a huge good interaction between the professor and the student. And some of you all know this, and if you have worked in our labs, and you know how things work. And there's a variety of research options you can do. So we just kind of want to go over them, and then I'll stop with that now that we have a panel that we want to start. So you can actually work on cancer surgery and ecology. And we've got the types, basically. That's pretty much what we're researching. You can work in research microbiota and DNA, and that's Dr. Janet William, who's an expert in DNA technology and DNA sequencing. You can work on yeast and protein interaction, especially for that heat shock proteins. Dr. Cote is basically, that's her degree and PhD degree actually is in. Or you can work in traumatic brain injuries and Alzheimer's, and that's pretty much what I work. And in addition to all of that, students can actually take modules to get a certificate in either cell culture or ELISA or DNA or protein purification. And these modules, they're about 3 weeks in regards to duration time, and you end up with getting a credit. In addition to that, you get a certificate and prove that you finished, a certificate program in one of these modules. And that actually is very helpful because that helps to build up your resume, and when you apply for a job, your resume is the one that's going to basically help you to get a job or not, and that's what we're trying to do. Okay. So all the information that you need is posted on the Elms website, the biotechnology website, or you can email Dr. Tice, you can email Dr. William, or email me, and we'll kind of be happy to help you out. Thank you.
I'm not sure if I actually introduced myself. So I'm Jay Marshall, Director of Career Services. I've been here for 2.5 years now. And it is my pleasure to introduce our panel tonight. Our emeritus panelist, who is returning tonight to be our moderator, Dr. Anthony Costa. He drives the development of ASO combination devices and gene therapy delivery devices at Biogen. He has over 25 years of medical device experience in areas of product development, project management, marketing, new business development, and technology scouting. We have, to my right, I think it's my right, yes, my right, Dr. Quentin Matthias. Did I do it right? Matthias. I'm right earlier. He is the Associate Professor of Nutritional Immunogenetics at the University of Connecticut. Before that, he was an instructor at Harvard Medical School and held adjunct positions at various institutions. He completed a postdoctoral fellowship at Boston Children's Hospital where he researched mast cell biology, allergic inflammation, and asthma. And in the center square, we have Dr. Sally Schneider, the director of HistoSpring, a core facility which handles histological needs for the region. She's an associate professor in the Department of Surgery at UMass Chan Medical School, Bay-State, and adjunct assistant professor in veterinary and animal sciences at the University of Massachusetts in Amherst. So please join me in welcoming our distinguished announcement.
All right, I'm sitting here at the table. Can everyone hear me? Okay. So if it's okay with everybody, I'm going to try and moderate this discussion, but I might be drawn into trying to answer a few questions as well too. So hopefully you'll indulge me and allow me to do that. This is a biotech panel. So let's start with what drew you to this particular career path?
Yeah, so I guess I can start. And, I was always very interested as a student in high school in the science realm. And when I was in school, I wanted to be a doctor. I'd read a lot of science fiction. Some of the books that I was reading were by Robin Cook. I don't know if any of you have read his books. I used to like some of his older books compared to some of the more recent ones. And he used to write medical thrillers. And that really got me very interested in research because he wrote, quite a bit about the latest advances in genetic technology, infectious diseases, vaccines, etc. And, I thought I really wanted to be a doctor and pursue something like this. But later on, as I went through college and things like that, I really started getting more interested in the research aspect of things. And that's actually what really led me towards biotechnology more than trying to pursue a career in medicine.
Excellent.
And very similarly, I always knew I liked science. When the teacher would be teaching, I'd be like, how do they know that? I always loved animals. I thought I wanted to be a vet, is what I thought. And in the end, I went to UMass Amherst for my PhD because I thought, I could go into the veterinary animal science program and maybe make my way over that direction. But I also love research, and I've always loved puzzle solving, and I see research as a lot of that. You find the puzzle pieces in the literature, and you start putting it together and trying to fill it in. And it just makes me very happy, and so in the end, I ended up staying in research, I guess more for monetary reasons. I could have gone to vet school, but I was like, they'll pay me to go to graduate school, and I don't want to be hundreds of thousands of dollars in debt. So I have my zoo at home, and I do research. But I had a lot of mentors that interacted with me through the way to help steer that direction of it.
Excellent. I'll just share with you guys real quickly. I may have shared part of the story last year. My undergraduate degree is electrical engineering. And I was a pretty good high school basketball player coming out of high school. I had planned to play in college. I was hoping to play in college. Started dislocating my shoulder the summer between my senior year of high school and freshman year of college. Over the next 3 to 4 years, I dislocated my shoulder about 10 times while I was in school. Every time I'd go into intramural basketball or something, I'd forget that my arm was injured and I'd dislocate my shoulder. One day, in the middle of my sophomore year, I had just dislocated my shoulder in intramural basketball. And I was kind of bummed out, walking around campus, a little bit depressed about it. I'm thinking, oh man, I can't play basketball for another 3 or 4 weeks. I'm going to get fat and sloppy, and all these things that were going through my mind. And someone rolled by me in a wheelchair, and the light bulb went on in my head. I said to myself, stupid, why are you upset that you can't use your arm for 3 or 4 weeks when here's somebody in a wheelchair who may never be able to use their legs for the rest of their life? So I started thinking about biomedical engineering, and I started to shift my electives to the biomedical track. Fast forward, I graduated, went on to grad school, got a master's in biomedical engineering, started worked for companies for literally 20 years, developing different medical devices, class 2 and class 3 devices, cardiovascular devices, urogynecological devices, and even more. And it took me that long to realize that there was an opportunity for my skill set in the biotech arena. So about 8 or 9 years ago, I transitioned to biotech for a few reasons. It gave me a new challenge because I felt that I had done everything I kind of wanted to do on the medical device side. And there was a huge need for biotech companies to have engineers because while they knew how to develop the drugs, they didn't know how to develop the devices to get the drugs into the body. So that's where I come in. I'm an engineer that's developing combination devices, and biotech companies are doing some incredible things, and I hope we can talk a little bit about that. And that's what really got me excited about the biotech arena. And then finding out about some of the new technologies that have come out, which I hope we can talk about today. So speaking of new technologies, what really excites you about different advancements in the biotech arena? What makes you go, wow, I wouldn't have thought we'd be going down that path 20 years ago when I first got into this?
Yeah, so I mean, that's a really good question. As an immunologist in particular, I'm very excited about all of the new immunotherapeutic drugs that are out there. I mean, Sally's a cancer biologist, and even about 30 years ago, most scientists and many immunologists thought the immune system probably had no role to play in cancer. But today we know that it does play a big role. And so there's a lot of different drugs out there that really target the immune system. And some of you have heard of drugs such as Keytruda and things like that, that have shown great efficacy at suppressing tumors and even curing some people. In terms of a real surprise, I think in the biotech industry, I think this whole idea of using mRNA as a sort of vehicle, not just for vaccination, but for various other purposes. I mean, I think that's probably been one of the biggest surprises in the last few years because just like with immunotherapeutic drugs for cancer, people have always been very skeptical about technology like that. And I think we've seen a rapid advancement, obviously, in the last few years since COVID.
I'm going to go more technology related. I'm really excited for single cell sequencing. And because 20 years ago, I started doing research on understanding how early pregnancy reduced the risk for breast cancer. And I would collect all these breast tissues from women and sequence, the transcriptomes, see what's happening. But there was so much noise because there's so many different types of cells in the breast tissue. And I'm totally jazzed. I haven't done it yet, but I'm very excited for what can show me in the future.
And how did your training in school or in the industry prepare you for the roles that you have now?
Yeah, so I mean, I did a bachelor's in microbiology and biochemistry, so very similar to what many of you are doing, and I ended up getting a master's in microbiology after that. But my passion was always for learning more about the immune system. I was very excited about immune cells and what they could do, so I really wanted to pursue a PhD in immunology. And I think over the years, I had the opportunity, just like you guys, from what I heard, learning many different types of techniques, especially molecular and cellular techniques, like ELISA, cell culture, Western blotting, all those kinds of things. I think really being involved in the lab with that and trying, like Sally hinted earlier, trying to solve puzzles. You really want to figure out what may be happening in a certain situation in a molecular cellular context. I think that really kind of is what you really need if you really want to do research. And I feel like that prepared me quite a bit. I mean, I had great mentors throughout my education, including my postdoctoral years. So I think mentorship plays a very big role as well.
Yeah, so I went to also a very small liberal arts school that's really focused on business and art, and there wasn't a whole lot of research, and that made me sad at the time. In the end, it worked out because I got to know the teachers really well. And the research project I did was in plant biology, which probably at that time would have been my least favorite type of biology. But one important thing I learned along the way is that's how much I love research, because it almost doesn't matter what I'm researching as long as it's the act of figuring things out. And then I took 3 years off to work as a technician and learned a bunch of techniques. And that's the next thing you realize, that technical toolbox you can take and use it in whatever direction you want. And in graduate school, I did not want to go into Barbara Osborne's lab because she was studying how cells died. And I thought, how boring. But that's when apoptosis was just coming into fruition, and I realized that, oh, actually, quite important and quite exciting. But it's funny how you can go in thinking it might not be that interesting, and it's all what you make out of it.
I think those are very good comments, and you guys talk about the research side of things and how working in labs allows you to develop those skill sets. I did research during my PhD in biomedical engineering, but I always wanted to go into industry and develop tools and devices to treat certain disease states. So the learnings that you will get through the biotech program can be leveraged in research if you want to do research. It could be leveraged for product development. One of the things that I think is really interesting is I work for Biotech company. Most people probably have heard of them. Many individuals who are biotech or biomed majors, they end up getting into process development, because when you're developing a drug, the first time you develop it, it may be just a few milliliters worth of the drug. You have to figure out how to scale that up to gallons, if not liters, and tens of liters, in order to provide a drug for a disease state that there's 1 million people a year who may need that. So those technical skills, biotechnology, engineering skills can be used on the process development side of it. So research is great. I enjoyed it, but my real passion was product development. And some people's passion can be process development as well. So we talked a little bit about your preparations in the biotech arena. What challenges do you see, or where do you see, what's the next great space for biotech to go into? We don't have the capabilities now, but if we had these capabilities, what would it allow us to do in the future?
That's a really good question. I think something you need to think about a little bit more. I mean, there's obviously in terms of disease, we've been looking at some diseases for a really long time, right? And it would be great to figure out maybe the holy grail of like trying to cure cancer, or even things such as aging and things like that. There's a lot of research being done in those areas. But I think where things, at least from my perspective, where I can see a lot of things going is the use of a lot of artificial intelligence in trying to perhaps develop new drugs or direct therapy in a certain direction. We've already gotten to the point where we're doing precision kind of biology in many different areas, whether it's genomics or nutrition, department I work in, things like that. So I think using all these new computational techniques that we have and artificial intelligence, I think will have the potential to really expand the scope of what we're able to do.
Dr. Snyder, where do you see the challenges?
A huge question. So I'm just going to narrow it down to my area of interest in cancer biology, and it's in personalized medicine and better diagnostics. And from what we do in my lab is building better models to be able to develop better precision medicine.
I was chatting with Dr. Matias right before this session, and we were talking about what we do in our jobs, and I had mentioned how, as an electrical and environmental engineer, I'm very impressed with the ability of biology and biochemistry to help reprogram cells for upregulation or downregulation of various proteins. And I was saying to him how I kind of explain that to people, because I'm not an expert in that area, but I say we now have the technology that allows us to reprogram cells, the nucleus, to produce different proteins, which is very similar to me writing a software program and having a microprocessor spit out new information or new code. And that to me is very impressive to see, to know that that capability is out there and seeing how companies are trying to leverage that. One of the drugs that I deal with is a drug by the name of Spinraza. It deals with a disease state called spinal muscular atrophy. And what happens is the spinal motor neurons are supposed to produce a particular protein to maintain their vitality. But people who have this, and what do you call it? There's one gene that's not working correctly. It's a monomeric disease. So they figured out what gene it is. They're able to use a drug to go in and send the right genetic code through an ASO to these cells, which causes these neurons in the spine to make the right protein. Before this drug came out in 2016, babies who were born with this disease, the doctors would tell the parents, take your child home and enjoy the rest of the time you have with them, because they typically didn't make it to their first or second birthday. Now, because of this drug, using these new techniques, people are able to have a much better quality of life. They're living longer. It's not a cure, but it's an improvement. And our ability to help the body figure out what it should do more of or do less of is really inspiring to me. It makes me think, if I was, and this is a question I have for you guys, if you were going to do things all over again, would you choose the same career path?
Yeah, before I get to that, I mean, what he was talking about is a wonderful example of personalized medicine. And, with the CRISPR-Cas9 and other gene editing technology we have now, it's something that's, right at the cusp, right? I mean, aside from asking a lot of ethical questions as to what needs to be done. The other things, some of the other things he mentioned too, scientists and labs are able to grow tissues, in culture and maybe even grow whole organs. So I mean, I think that's a pretty big thing. We're using a lot of 3D technology now that was never possible before. And there's been a lot more research lately, and you were talking about some of this, trying to somehow harness the neural circuit by implanting devices and things like that, that could then maybe help you control limbs and other organs and things like that, especially for people, who may not be able to do that due to injury or some sort of genetic deficiency and things like that. So in terms of whether I would choose a different career path, I'm really excited and happy about my specific career path in terms of the passion and things like that. In terms of challenges, I'm sure you're going to ask us that as well later on. Research can be very challenging, and it may not, or depending on where you end up, if you're in academics, it may not always be monetarily fulfilling. So there's, but in terms of my own passion and career path, yeah, that's something I would definitely choose to do.
Yeah, exactly what I was meant to be. I
That's a good
mean,
answer.
what was the exact position I have right now, what I was aiming for? No, but it goes to my ability to adapt to the situation based on my needs and wants. I wanted to have a family. I wanted to do it both, science and family, and that it's easier in Western Mass and it's easier here than it is in Boston. So that is, yeah, no, absolutely. So,
I think for me, I probably would have switched to biotech about 10 years sooner. Part of the reason why I got into medical devices is because I wanted to help people and improve the quality of life. But I started consulting for Biogen back in 2020, right before COVID. And I was working on aducanumab, which was the first Alzheimer's drug approved in the last, well, I guess ever, right? The first one that was approved. And I was working on that project. I was working on the combination devices to get it into the body. And I realized that we're dealing with about 6 million people in the United States who are dealing with Alzheimer's who could potentially use that product I was working on. So it gave me an opportunity of getting a much broader reach of the work that I was doing to affect a much larger number of people. But I think, like these two, I've been fortunate that I'm doing the things that I like to do, which is device development. I'm doing it in a biotech arena now, but it's similar to what I had been doing my whole career. So I don't think I would necessarily change career paths. I may have made the switch to biotech a little sooner. I can tell you one thing. I was talking to my dad as I was riding up here today. There's a different compensation scale between biotech and medical devices. I don't know how many people know that, but yeah, biotech pays better than medical devices. One thing you said made me think about something I heard a few months ago, and this may have been a play in immunology, or it may have been genetic manipulation. But wasn't a pig heart or a pig kidney implanted into a person? And what did they have to do to modify that pig organ in order for it to not be attacked by the immune system?
Right. So I think we heard about that a few months ago, and I can't remember exactly what they did with the pig heart itself, but definitely for the recipient to receive something like that, because that's considered a xenograft, which is you're getting a tissue from a completely foreign individual. And your immune system's normally going to reject something like that. So you really have to be able to suppress the immune system to a specific level. So patients are typically on a whole regimen of immunosuppressive drugs. There's certain organs that are easier to transplant maybe than others. But getting an organ from a completely foreign organism, such as the pig, you'd have to modify some of the antigens on that pig heart so that the immune system would at least get a little bit tolerant of some of those antigens. And I think, unfortunately, while it did expand the person's life for a few months, it was not totally successful, if I'm correct. And so I think there's a lot more that definitely needs to be done to achieve success in that area.
By a show of hands, how many people have heard of CAR T? 1, 2, This is another area of biotech that I find fascinating. Do you want to explain to them?
You want it? It's your area.
Whose area is that?
All right, so CAR T cells are, based, what we call them CAR T cells, right? And the CAR stands for chimeric antigen receptors. And so basically, what you do is every T cell, which is an immune cell, an adaptive immune cell, expresses a receptor on its surface that's very specific for a particular antigen. And one of the problems we've had with tumors is because they're the body's own cells, right? Our immune cells get tolerant to these tumors, and so they don't really attack the tumors as well. And so what if you could kind of harness some of the latest technology we have now and render these T cells that we have more hyper-aggressive against these tumors. And so one way scientists did this was by engineering a molecule that they call the CAR molecule that these T cells could now express that not only had specificity for that specific tumor antigen. I mean, I know I'm going a little bit deeper into some of this biological stuff, but also gave them these other properties that significantly enhanced the activation of those T cells. And they were also able to produce certain proteins that made them much better at proliferating and killing the tumor cells. So without going into too much detail as to what those individual components are, there were 3 different protein molecules that were brought together to make one specific receptor that these cells could now express in addition to the receptor that they already had that could recognize the tumor antigen. I don't know if any of that made sense, but it's pretty cool technology to be able to do that. First used to treat leukemias, and we're looking at obviously using this type of therapy for a lot of other types of cancers.
One of the reasons why I brought that up is because that was a very interesting technology that I was exposed to. And when I first got exposed to the technology, I was told it costs about $500,000 to extract the cells from someone, help them proliferate, and then being able to put them back into the body once you have inserted new genetic material into them, right? So they said $500,000, and it takes about 30 days for that to happen, or to process the cells, which means that it's too costly for most people to have that particular therapy. I used to be the head of medical devices for Draper. Draper they're known as a defense company, but they have been developing great technologies for 50 to 60 years, and we were looking to leverage some of that technology for medical device applications. And one of the things that we did is, I didn't actually do it, it was a team of other engineers. They combined biochemists, mechanical engineers, electrical engineers, chemical engineers, and they figured out a way to transfect the cells very efficiently. They could do them one at a time, put them in there was a flow cell where the cells would go through one at a time. They could transfect them. They exposed the cells to a transient electric field where it made the cell membranes permeable. The new genetic material could get put inside there, and then they had a specialized way of helping those cells proliferate. And it turned out, using engineering, biotechnology, they were able to take a procedure that took 30 days and cost $500,000 down to something that was about $50,000, and it took 3 days. So they had prototyped this, but it took biotechnical engineers, biomedical engineers, electrical engineers. All of these disciplines had to come together to solve a problem that people didn't even know really was a problem. They just assumed, oh, this is how we have to do it. And they basically said, nope, we're not going to do it that way. If we had to do it from scratch, how would we do it differently? So I say that to let you guys know that even though there may seem like there's limitations to what technology can do, all you need is that breakthrough idea and looking at something from a different perspective to go down a pathway that could be hugely beneficial for the industry and for patients. So don't underestimate what you have up here.
And that's a very good point. I mean, I think all of us would agree, what's the big thing about research? It's asking questions. And you, the more questions you ask, and you're thinking, what's the next big question in this field? It's kind of what you asked before. And how can I, if I have the expertise or whatever, how can I possibly help solve that question? And so, like Sally mentioned earlier on, it's all puzzle solving, and you really got to be asking the questions to drive you forward towards the next step. And that can be exciting when you're making even those little discoveries along the way. And that's what drives the passion in the end. There's a lot of times when you're doing research, and we've heard this over and over, right? I mean, Edison tried to find out, what would turn that bulb on, like over 100 times, and I'm sure he was frustrated, but he kept going on. So, and I think you'll always have those challenges, but eventually, hopefully, you'll figure out how to answer that specific question.
So Jay, I could keep peppering questions to the panel. Do we have time to see if there's questions from the audience?
We do.
Are there any questions from the audience? I want to make sure we don't miss an opportunity to answer any burning questions that you have. If not, I get to keep peppering them with questions. So
He asked us the hard ones.
We have a question over here.
yes.
My question kind of like goes back a little bit about the pig heart transplant. Knowing how sensitive the human body is, even like different blood types, was that one of the reasons why it might have been rejected because of the different blood types?
Yes, I mean, definitely the blood types do play a big role, but even more so than that, all of us as humans, we express these specific antigens, and I don't want to go into the details of that, but it's called MHC antigens, and these genes directly coordinate how our T cells and our immune cells would respond to specific antigens that are out there in the environment. And so as humans, each of us expresses a very different profile of these MHC antigens that unless you're an identical twin and you're totally genetically homogeneous, none of us are going to have that same MHC profile. And so aside from just the blood transfusion type, that's one of the reasons why it would be very difficult for me to just donate an organ to one of you sitting over here, unless you could find some compatibility between the two of us, and then the other person would have to go on immunosuppressive drugs. I don't know if that helps answer the question. Okay.
Well, I was going to answer the question because the pigs actually produce a sugar. And so exactly what you're saying, the human body reacts to that sugar. But pigs specifically, so pigs were sort of identified because of the size of the organs are compatible, but they produce a sugar that we don't produce. And so our body sees that sugar and reacts, very negatively to it. So they genetically modified. They basically deleted that sugar molecule in these genetically modified pigs specifically so that they would be organ donors. The X-style.
Oh, okay.
Yeah.
That's right. Yeah. Now I remember. Yeah.
Simon?
So you talked about two things that I found pretty interesting as the oncolytic viruses and CAR T-cell therapy. I was wondering if those are usually just like therapies that are given on their own, or if those are combined with other things like a multi-level therapy, like oncolytic virus and a CAR T cell therapy? Is that something that is on?
She's a cancer, you know.
Yeah, it's still out of my area of expertise, but I don't know if they do them together.
I think, I don't know about those two therapies specifically, but immunotherapy right now is not a standalone therapy. So they, at least as far as I know, I mean, you still do it in combination with chemotherapy, uh, if necessary, radiation and things like that. And I'm not sure if there's any disease where just immunotherapy alone. I mean, well, I think in the case of leukemias and things like that, I mean, bone marrow transplantation, hematopoietic stem cell transplantation is known to be an efficacious therapy all by itself. Uh, but CAR T cells, I think, would be a different uh, uh, kind of therapeutic regimen.
Yeah, my question is a little more broad and just, I was wondering if you could share some advice for someone in my position who's looking to enter this.
What is your position? It's
I
like,
mean, like graduate student, like just anyone surface level new in the biotechnology.
oh, you're in the master's program,
Yes.
biotechnology? Okay.
Yeah.
I would say get as much experience. I volunteered in several labs, and I found that I was viewed very differently coming in, having done that. People are like, oh, they have experience. Oh, she's willing to go above and beyond, not just for money. So it definitely leveraged me up, and getting whatever internship or experience you can get. All welcome.
I think for myself, it's funny, I've been having this conversation with my kids and some of the people that I mentor. I look at school very differently now than when I was in school. When I was in school, it was all about learning what I need to learn for the exam, get through, get good grades, go out and get a job. I'm telling my kids who are now, one's in grad school, one just started their freshman year, I'm telling them, learn as much as you can from your professors and from your schooling, but look for a problem that's important to you that you want to solve. And use that education that you're learning to figure out how to solve that problem. Because when I was 18, 19 years old, no one was talking to me about entrepreneurship. And there are so many opportunities in the biotech arena and the medical device arena. If you see a problem and you come up with a good solution for that problem, you may be able to start a career by working in that space. And somebody may want to buy that solution from you. So learning for learning's sake is important, but I think learning that material in order to apply it to a problem just makes that information stick in so much better, and it makes you a better engineer, better scientist, better whatever.
Yeah, and I think from the practical point of view, you really have to ask yourself, what is it you really like? What is it you really like to do in the future? I mean, there are a lot of students I know who've gotten a master's, for example, and then have gone into the biotech world, worked in pharmaceutical industry and things like that, and found maybe positions such as research assistants and things like that. But if you really want to be a driver in the field, where you're just not, simply doing a job in a lab as a research assistant to something, I mean, you probably want to consider getting a PhD, and then if you want to go into the biotech world, you can get into R&D and things like that, where you now have your own lab and be driving some of the research in that area. So I think you really have to consider whether that's the next step for you, or like Sally said, if you just want to get some lab research experience and then just apply for jobs in industry, I mean, that's another path that students also often take or choose to do.
One quick comment, and then I think there was a comment over here. something you said resonated. Dr. Schneider said, go in the lab, try some things. At this point in your career, try everything. Anything that looks interesting to you, try it out. You may find that you have a passion for it. You may find that you hate it. But that's important information, because then you just take that off your list of things you want to do. If it's something that you hate. But I think the more you can expose yourself to, that may help you become more well-rounded, and it may help you to find what you're passionate about. My passion didn't come out until something was taken away from me, and I saw that I could use my knowledge in a different area. Who knows what's going to, when that light bulb is going to go on in your mind to say, this is what I want to do, and I have to do this for the next 20 or 30 years, I'll be happy doing it. You know? I'm sorry, I told you where you had a.
Yeah, just one thing. That was the reason why we made these modules. The idea of having a subculture module and a larger module, protein purification here, is to help you first get experience, second, build up your resume. And these are two things that when you apply for a job, you will be standing out, you know, you will be competitive. And actually, it has been proven today. I was talking to Jay about one of our students, Brendan, has basically is taking 2 modules with us. And he was asked for an interview at Agilent. And he went, and we're waiting actually for him to, you know, hopefully he can get a job, you know. So that is the whole idea of us making the modules to have you, you know, build up your resume and kind of stand out with that job.
Earlier in our conversation, Dr. Matthias, I think you mentioned mentorship. And that's one of the questions that I had. How important is it for you, Dr. Schneider, to have mentorship, and how important do you feel it is for these younger people at the earlier parts of their career?
Extremely important. I would say throughout your career, even now, there are people I look up to, I will talk to. Sally has been a mentor to me in some ways. It's interesting. Neither of us knew we were both going to be on the same panel, and we worked together for the last 50 years. And so you can have this kind of peer mentorship too, you know. But I'd say it's definitely it was definitely very critical to me. And you can have different kind of mentors, and some people, their style could end up totally, can be totally off-putting. So I'll give you an example. And this is something I wanted to bring up earlier too. Sometimes you go into research thinking, oh, this is so exciting, I want to do all these great things. And, you know, almost, when I grew up, almost everybody around me wanted to either cure cancer or AIDS. And I was like, oh, I'm going to get into the research lab and start doing that. And the first lab I got into was a cancer lab, and I worked there for about 1.5 years. And my mentor in that lab, I mean, he had great ideas, but he didn't work very well with students, and there was almost some kind of exploitation in a way, like going on in that lab, where people just worked all the time, and there were a lot of negative comments, and you know, you never succeed. This area is never going to go a certain way or whatever, and it really made me question. You know, what am I doing here? Do I really want to do research? It's kind of like what you said. You might think, oh, you wanted to cure cancer, but you're just trying to figure out this ELISA. It's taking forever, right? And so, but then subsequently joined another lab and had a different kind of mentor. He was a great person, and let me do things all by myself, but he was never approachable, never really had the time to talk about research and things like that. But he taught me a lot of things, you know, that I'm grateful for to this day. So I think throughout your career, you'll have different people, and everybody has a different style. but it's extremely important to learn from them, and made a big difference for me.
I completely agree. I had an amazing undergraduate mentor. I had an amazing graduate mentor. In between there, I worked at MIT in several labs. One was a Nobel Prize winner, and he taught me how I absolutely do not want to be as a mentor. The one I went to after that, also at MIT, again, another good example of how I do not want to be as a mentor. But all very important, right?
Yeah.
And you're right, they're all different, and every student requires something different. So not all students will do well with me. I responded really well to those that were very encouraging and challenged me to do things on my own, but would not let me totally drown, would not ignore me, was always there with the door open if I had questions, but really wanted me, pushed me. I responded well. I didn't respond so well to the micromanaging. Definitely did not respond very well to the lab where they put everyone on the same project to compete with each other. Yeah, you learn a lot working in all those different situations about how you want to do them and how you are to your students.
I think mentors are extremely important. And I look back on my career, and I've had a few mentors along the way. Some were actually really good, but I didn't have enough of them consistently. And by the time I started getting mentors, I had bumped my head into the wall dozens of times. And I'm talking to my mentor, 5 years later, I'm like, if I had known that 5 years ago, I wouldn't have run into that problem in this situation or that situation. So one of the things I do now, I have several mentees, and I try and be for them the mentor that I wish I had when I was in my 20s and in my 30s. And what's interesting, I just saw one of my mentees yesterday. She was presenting at an optical conference at MIT yesterday. She's in a PhD program at Columbia, electrical engineer. And I met her in her sophomore year. She came from UMass Lowell. And she told me that she was interested in cancer research. I was like, wow, that's great. And she said, but I can't do it. I said, well, why can't you do it? She said, I talked to one of my faculty members, and he said, I'm an electrical engineer. I can't do cancer research, so I'm going to get a master's in acoustics. And I'm looking at her, I was like, who would tell you such a thing? And she said, I went and talked to my advisor, and he said, no, you're an electrical engineer. You can't do cancer research. Electrical engineers don't do cancer research. And then I told her about what I shared with you guys about the work that was going on in Draper with CAR-T technology and how electrical engineers were part of that process. And she ended up getting her master's degree in acoustics. I told her about a few PhD programs. She got into Columbia University. She is working with a world-renowned professor who is doing what? Cancer research. Her presentation yesterday was OCT, optical coherent tomography. She is doing high-resolution scans of breast tissue in order to find which breast tissue is cancerous. And here she is giving a very good technical presentation amongst her peers, colleagues, other professors. And I'm thinking to myself, this was a young lady that somebody told 5-6 years ago that she couldn't do this kind of research. What does she want to do? Cancer research, open up her own lab and have her own students and really just focus on cancer. So I went off on that tangent to say, and I'm kind of piggybacking on what we heard here, not every person that comes into your life is going to give you good information. So you're going to have to be intelligent enough to kind of distinguish, is this person giving me good information or are they maybe not giving me so good information? And there's never a problem of having more than one mentor. And you can ask multiple people the same question to see what type of feedback you get. And it's your job to determine which way you want to go. I tell my mentees, my role as a mentor is to help you figure out the options that you have. I tell them the choice is yours. I'm not going to tell you what to do, but I want to make sure you've considered the ramifications of whatever decision that you make. And that's what I wish I had when I was growing up or coming up through industry. The last thing I will say about mentors, unless there's a specific question, you as a mentee, it's your responsibility to maintain the relationship with the mentor. That's where I fell awry because I wasn't as consistent with my engagement with them. And they're busy people. You know, they were 10, 15, 20 years ahead of me in the industry. They're not sitting around waiting for me to call them, you know. So if you find a mentor, someone who wants to work with you, make sure you do your part to maintain that relationship. And I think it will be very valuable.
Okay.
Any other questions before I start firing?
I actually have a question. What advice would the panel have for biomed, biotech students who are looking to graduate, go out into the Pioneer Valley specifically, this region? Where do you see the opportunities? Are there any companies that you think would be great places to knock on that door? Or is it more of something where they need to seriously think about going east?
There is not a lot out this way, unfortunately. We used to rent, Middle Four Sigma expanded in our facility, and I would have said that was great. There were a lot of engineers doing, and then they came in one day and fired everyone. So, and we've been trying to get more companies to come use our space because it's a great place to live and so much better than Boston, so much cheaper than Boston. But they all want to be in Boston. So it's harder.
I mean, what I'd say is, if you're specifically looking for biotech industry, it's going to be a lot harder. But you know, we do have two big universities around here, UMass, UConn. There's a lot of other colleges and liberal arts universities where faculty are doing research, and there's probably always the opportunity, like Sally was saying, to find a research lab and get some more expertise before you could potentially, you know, maybe head to a big company or something like that. That said, I mean, if somebody's willing to travel, I mean, AbbVie's in Worcester, so you know, there are a couple of small biotech companies around there, and there's Boehringer and Bristol Myers Squibb, maybe about an hour from here, or hour and a half. So if somebody's wanting to travel, you know, there are some of those companies. Pfizer, of course. I mean, yeah, you have that's far enough actually. That's in Groningen. Pfizer's in Groningen, and then it's an hour and a half to Cambridge. So, depending on how long you want to be on the mass bike, or maybe you want to ask some of our legislators to get that high-speed rail going.
2 hours of time drive.
If you would have asked me this question 5 years ago, I probably would have had a very different answer. And the reason why, my answer 5 years ago would be, and I don't know very much about the Pioneer Valley because I'm on the other side of the state, but there is so much activity in the Boston, Cambridge area, biotech, medical devices, that I would say, if that's your field, you may want to go a lot closer to Boston. But since COVID, one of the benefits of COVID is that companies got used to employees working remotely. Most of the people in my group are remote. I only go into the office one day a week, maybe twice a week, unless there's big meetings. Now, if you're an individual who has to work in the lab, you may have to go into the office more often. I have a colleague, she is a biochemist, so she's one of the people working in the labs. She moved to Framingham, actually a town a little bit on the other side of Framingham. So she's not very, very close to Cambridge, but she only has to go into the office 3 days a week. So now companies are much more receptive to the idea of people working remotely in whatever capacity you are in the organization. So if it's my first job, I would probably be, if it's a good company, I would go wherever that company is. That company could be in Alaska. If it's my first job and it's a good company and I like what they're doing, I'm going. But that company may not force you to be on site every day. So you may have more flexibility. So I would say don't totally not look in the Boston area, but if you do go that route, try and negotiate time, you know, working from home.
Right, and we're lucky actually to be in this area. I mean, we have a lot more choices compared to somebody in, maybe Idaho or, somewhere else. So it's actually not that far. I have a student of mine who just graduated recently. She was in Connecticut and she got a job at MIT as a lab manager. She's been traveling every day. I told her, I don't think you're going to do this after a year. You need to find a place closer to Boston, but she's still been doing it. So I think it's doable, like he said, especially if you could put in a couple of remote days here and there.
And that being said, at UMass, there's a whole innovation institute, and they're trying to start all these small companies. They aren't as maybe secure as your Biogens or things like that, but that's an opportunity, right? Where you learn things and you might get in the ground floor. Entrepreneurship as well as research.
Especially for your first job, and then always be.
That's a great point. I wouldn't underestimate an opportunity to go to a startup. Because startups don't have all the resources as the big companies, but they don't have all the bureaucracy either. And a lot of times what happens in the startups, I had a mentee who was asking me, should I go to this startup or should I go to, it might have been Bristol Myers Squibb, she got a job at. And I said, well, aren't you working for them in the summertime? She's like, yeah. Did you like what you were doing? Yeah. Were you wearing a lot of hats? Did you get exposed to a lot of different things? She's like, yeah, I really did. I said to her, I said, if you spend 3 years in that startup, that's probably the equivalent of 5 to 7 years in corporate America because you're very regimented to a particular area. Startups don't have a lot of people, so they can't say, oh, you only do this. They make you do this. So you get exposed to so much more quicker in a startup. You may not have the confidence of them being around for the next 5 years because startups are difficult. But I think the opportunity to learn from them is tremendous. And in some respects, I wish I had spent some time in a startup earlier in my career. I waited until I was 50 to start my own startup, I think.
Just to repeat that, you said start your own startup?
Start my own startup, yes. How are we on time? I never did the time check.
So we're at 6:15. Maybe we should switch to prizes and then maybe some shifting around the yes.
Can I ask one more question?
You absolutely can.
A closeout question. So what words of wisdom would you want to leave to these students who have been interested in the biotech and, what have you learned over your career that you said, you're saying, I wish somebody would have told me this. What would you want to leave, impart on this group here?
Something new, it's so I can think about it. I think it's not anything I haven't already said, which is get all the experience you can get. So take it all in. Don't close any of those windows yet.
Right. And, I would say definitely follow your passion, follow what excites you. And then along with that, take practical steps to see what you actually want to do. And, my career path may not be the same for you, and it's going to be different for everybody. You might find one aspect of biotechnology that excites you more than others, or a certain kind of profession. And these days, I mean, we have this interdisciplinary stuff going on where all of these different fields are intersecting together. There's so much you can do. Electrical engineering, in computational sciences, et cetera, et cetera. There's so much you can do. So look at your strengths, look at what you like, try to find opportunities where you can get some experience and practice. And one of the things my PhD mentor told me that's always stuck with me, especially for those of you reading any hardcore research or working in the lab, there's no such thing as good data or bad data. It's always good experiments or bad experiments. Are you designing a good research question? Are you designing a good experiment? And then whatever data you get, follow the data. So I think that's really important. A lot of times we can kind of get biased because we think things have to be a certain way, but they may not be, and that's where oftentimes the new discoveries come up.
Last thing I would say on that topic is don't be afraid to try new things, learn new skill sets, stretch yourself. Don't be afraid to step into that uncomfortable space, because that's where you challenge yourself, and you may find something surprising about yourself. And then the other thing I would say is you've got great resources at this college. Make sure you leverage those resources as much as possible. You may have a need to study something that's not in your core curriculum, but there's faculty here who may have that knowledge. And what I've found is most times faculty, and I'm one of them I'm an adjunct faculty at school. Most people love to talk about the work that they do. And very rarely will you run into a faculty member who probably won't give you time if you approach them about something that they have passion about. So it's a great way to get to know them. It's a great way to get insight into skill sets that you may want to acquire. So just make sure you leverage the resources that you have here.
Excellent. So, round of applause for our panel. And before I do the prizes, I do want to impose to get a picture. Dr. Thomas, Dr. do you mind joining us? And on the table. So,
Shall we sit here or?
please.
Okay. You don't need to.
Sanders.
Make sure you get my good side.
Is he good with the?
So children can
They didn't throw it to me.
Thanks for you.
Don't worry. Thank you. And before I do the prizes, I am going to remember this time to end the Zoom because last time I kept people hostage for another hour. So thank you for joining us.